welcome to my channel. This is Ace with IGCSEs with Clarissa, and today we'll be solving January 2023 Edexcel Biology past papers. And yeah, we will begin with question 1. Okay. The diagram shows a flower from an apple tree with some structures labeled. We have a flower here, and it has some labeled structures: U, V, P, Q, S, T, and R. Okay. It's asking for the first question asks, this apple tree is able to self-pollinate. Which is the correct transfer of pollen during pollination? Okay, so basically it says that this tree is able to pollinate with itself, like it doesn't need to do cross-pollination. So how does the pollen where is the pollen transferred from in pollination? So the correct answer would be C, which is V to P. V is the anthers and P is the stigma. As we know, in pollination, the pollen grains are transferred from the anthers of a plant to the stigma of the same plant. This is self-pollination. Cross-pollination is when the pollen grains are transferred from an anther of a certain flower to the stigma of a different flower. So our answer for the first question is going to be C. Which structure does the pollen tube grow down? So once pollination has occurred, obviously we just said that the pollen grains land on the stigma. So when the pollen grains land on the stigma, they start sort of like digging a hole into it and making a tube. This tube is going to act as a pathway for the pollen grains to get into the ovaries. So that structure would be Q, which is the style. As you can see here in this diagram, it says Q and it's pointing to the style, which is sort of like the stem of the stigma. Okay, which structure develops into a seed? Okay, the structure that develops into the seed is T, which is the ovule. This is the ovary, and then inside the ovary we have like small egg sacs. They're called ovules, and this is going to be C. Okay, the flower from an apple tree is insect pollinated. What is an insect pollinated flower? It's basically a flower that gets its pollen grains by insects. So let's say, for example, we have flower A. Okay, flower A anthers they made pollen. Okay, so we have a bee, and this bee landed on the flower to drink nectar. So it landed on the flower, it was drinking the nectar, and as it's leaving the flower, some of the pollen grains get attached to this bee. Okay, this bee then moves on to another flower or another plant, and it also lands on it to drink its nectar. So these pollen grains fall onto this other plant. So this means that this plant was insect pollinated, which basically means that the pollen grains were transferred from one plant to another by an insect. It says give 3 differences between the structure of this apple flower and the structure of a wind pollinated flower such as grass. So what are the differences in the flower and the structure of the flower itself in an insect pollinated flower and a wind pollinated flower? And a wind pollinated flower is a flower that gets its pollen grains, or pollination, as we just stated, is the transfer of pollen grains from the anthers to the stigma. That can be in the same plant, which is self-pollination, or it can be into 2 different plants, which is cross-pollination. But my point here is, how would the structure of an insect pollinated flower differ from the structure of a wind pollinated flower, which gets its pollen transferred by wind? There's no third party here. There's no insect that carries the pollen from one place to another. The wind just carries the pollen. So how would the structure differ? First thing, for the insect pollinated flowers, or the apple flower in this scenario, the petals of the flower are so much bigger than in the wind pollinated flower, and also they are brighter. They have they're colored, because most wind pollinated flowers, they have green petals, but for the insect pollinated ones, this is a different case. They have bright colored petals. Also, the stigma, which we said receives the pollen and pollination, is sticky in the wind. In the insect pollinated flowers, the stigma itself is sticky, so that when the insect lands on the flower, the pollen sort of sticks to the stigma. It also has nectar. Most insect pollinated flowers are nectary, they contain nectar, unlike the wind pollinated kinds. And also, their pollen is very large and it's very sticky, so that it can stick to the body of the insects carrying it. Yeah, like you said, it's asking us here for 3 differences, so we're just gonna state 3 of all the things we just said. So I'm gonna go ahead and say large petals and bright colored petals, and I'm gonna go with nectary. Okay, moving on. Okay, question or part C says the seeds of many plants are surrounded by sweet tasting fruit. Suggest how this enables the plant to spread their seeds. So many of the seeds of these plants are surrounded by the fruit that we eat, because whenever you're eating any sort of fruit, there's always a seed in the bottom, or sorry, not in the bottom, in the middle. For example, in a peach, an apple, there's always seed in the middle. The question is asking you how this helps the plant spread its seed. So how would having a fruit around the seed help it in spreading? So when the seed has a fruit around it, this fruit can be eaten. It can be eaten by animals, it can be eaten by people, it can be eaten by insects. Anything can eat that fruit. So when the fruit is eaten, the seed is then thrown away. Obviously, people are not going to eat the seed, so we throw them away, we disperse them. Or maybe birds or animals consume them and they release them in feces. So this sort of helps spread the seeds around, instead of, like, if they had no fruit around them, nobody would pick up the seeds and throw them away, or release them with feces. So this enables the seed to be moved around. So our answer here is going to be that the fruits are eaten by people, animals, and then the seeds are moved to another area. As we just said, they're thrown away or they're released in feces. Yeah, you could also phrase this differently. You could always say that it was released in feces, or that it's thrown away, whatever suits you. Okay, now we're done with question 1. Moving on to question 2. Okay, the diagram shows a transverse section through a human heart. It's showing us a section or transverse section through a heart of a human. Here we have a couple of labeled structures. We have the coronary vein, we have the coronary artery, left ventricle wall, right ventricle wall. Draw an X on the diagram to show the position of the septum. What is the septum? A septum is the wall between it's like sort of like a wall that separates the right side of the heart from the left side of the heart. So here in this diagram, our septum is going to be in this area here in the middle. It's going to be that wall, that thick muscular wall that's dividing the right and the left side of our hearts, and you're just going to label that X as it asks us in the question. Okay, a very easy mark to get. State two differences between the composition of the blood in the coronary artery and the composition of the blood in the coronary vein. So we want to know the differences between the blood in the coronary arteries and in the coronary veins. So the coronary artery is the blood going to the heart muscle itself. As we know, coronary arteries, they supply the heart, right? So this is the blood going to the heart muscle. Okay, the heart muscle is the pump that pumps the blood around your body. So obviously that blood going to feed the muscles of the heart is going to be oxygenated blood. It's going to have a high oxygen content because we want to do aerobic respiration, because our heart is a muscle and we want it to keep pumping. So obviously the blood that is going to get to it needs to be fully oxygenated, or it needs to have high oxygen content. And it also has less carbon dioxide because we don't want to increase the pH at the heart. We could also say that the coronary veins contain more carbon dioxide compared to the arteries, because they're going to take the blood that's coming from the muscle. We use the blood that came from the arteries that was fully oxygenated, and now we want to get rid of that excess carbon dioxide, so we just send it back into the veins. So we're gonna say that the coronary arteries contains more oxygen, and it also contains less carbon dioxide, but you can also say that the vein has or contains more carbon dioxide. I'm gonna go with the vein. Okay. Explain the differences between the left ventricle wall and the right ventricle wall. Okay, in a question like this, if you have no idea what to say, and if you're stuck on a question that you're saying, have no idea what to say, here in the question, it gave us a diagram that can sort of help us to get a mark or two. So first of all, if you look at this diagram that you have in front of you, you're gonna realize that the left ventricular wall is so much thicker than the right. This is, if you don't know anything, if you have no idea what the answer for this question could be, you can just look at the diagram that it gave you, and you might get a mark or two. So if we look at this diagram, we can tell that the left wall is significantly thicker than the right one, right? As we can see that right here. And also it's so much thicker than the right ventricular wall, so that means that it has more muscle or more muscular tissue, because we just established that the heart is muscle. So if it has a thicker wall, then it has more muscular tissue. Okay, why would the left ventricular wall need more muscular tissue, or why is the wall thicker? Because it needs to generate more pressure, because as we know from cardiology, is that the left side of the heart pumps blood all around the body, while as the right side of the heart just pumps blood to the lungs. So in order to pump blood to all parts of the body, you would need to generate more pressure than you would on the right side. So we're gonna phrase that right now. So we're gonna say left wall is much thicker than the right wall. Okay, so it has more muscular tissue to enable it to generate more pressure parts of the body. Okay, so overall the questions are pretty easy, nothing so hard. Okay, moving on. Part B of this question, the development of the coronary heart diseases is linked to a number of factors. Explain how these factors can increase the risk of developing a CHD or a coronary heart disease. So it says that developing coronary heart disease is linked to multiple factors, as any disease on the face of the earth. And we want to explain how certain factors can increase the risk of developing a coronary heart disease. So one of the factors that I can think of is smoking. For example, smoking is a very high risk factor for CHD, as we all know that smoking has certain certain contents that increase blood pressure, and so increasing blood pressure increases the risk of more clots. Also, high blood pressure in general is a very, very important factor that increases the risk of CHD. We have stress, because as we all know, stress raises the heart pressure. Stress raises blood pressure. Also, if obesity increases the risk or increases the strain on the heart. As well as if you consume a high fat diet, if you consume a lot of fats, have a high cholesterol diet, which blocks the coronary artery walls. Lack of exercise as well, because exercise, as we all know, exercise reduces blood pressure and it also strengthens your heart muscle. So lack of exercise reduces all of these. And also it could be genetics, it could be something, maybe it's an inherited factor. Yeah, okay, so we need 4 points. We're gonna go ahead and write those. So high blood pressure puts on the heart high fat diet, blocks Okay, this is the end of question 2. It was a total of 10 marks, and we're gonna move on now. For question 3, the diagram shows the nutritional content of 2 non-dairy milk products, oat milk and almond milk. We have 2 kinds of milk here. We have oat milk and almond milk, and we have the nutritional value. We have the energy content, the fat, the sodium content, carbohydrate, fiber, and protein for both kinds of milks. And a person is told by their doctor that they need to lose weight. Use the information from the milk contents and your own knowledge to discuss which milk would be most suitable for this person. So here we have both kinds of milks, and we want to know which would be more suitable for a person that wants to lose weight. So first of all, we are gonna look at the energy values for both of them. And as we know, the higher the energy, the less the weight loss. So first of all, when we're picking the milk, we want to look at the energy, and we want to look at which of them provides less energy for the same amount of milk. So if we look at here, we have oat milk, it's 502 kilojoules, and for almond milk, it's 251. So obviously, almond milk is a better option energy-wise. Okay, and then we're gonna also look at the fat content, the carbohydrate, the sugar, and also the protein, because protein is very, very good for you. So for the fat content, as you can see, almond milk is so much better. We have, for oat milk, we have 5 grams, almond milk 2.5 grams, which is so much less. Same amount of saturated fat for both of them, not much of a difference. Higher sodium for the almond milk, but that's irrelevant to weight loss. Carbohydrate, we have, as you can see, less grams of carbohydrate for the almond milk, but we also have the same amount of sugar for both of them. Less fiber for almond milk, which is not good. Obviously, you like more fiber, as it reduces the risk of constipation. So here the oat milk gets a bonus, and it also has more protein, which is also a bonus. We want to balance out our diet. It provides more protein, which we need for growth. So first of all, we're just gonna state everything we just said. So we're gonna say that oat milk provides, I don't know why this iPad is acting up, provides more energy. So more energy, less weight loss. Also, oat provides more fat. We don't want that as well. And more carbohydrates. And we're gonna state both of them, so that is all the disadvantages. We're gonna say that oat milk provides more protein and fiber, which prevent constipation. And we're also going to state that we can also say that oat provides the same amount of sugar, like almond and oat milk have the similar amount of total sugar. So similar sugar. So personally, you can make this conclusion. You don't really need to. You're not required to do that. But you can do that at the end. I'm gonna do that. So personally, I think they should consume almond milk. Okay, this is it for question 3A. Moving on, we have suggest why a person might drink a non-dairy milk such as oat or almond milk rather than cow's milk. Why would a person drink oat or almond milk when they can just drink normal cow or dairy milk? So there's a bunch of reasons. Maybe that person is lactose intolerant, which is a very common thing. Some people just can't handle lactose because they don't have the enzyme lactase that breaks it down, so they can't drink dairy milk, so they have to opt for the non-dairy options. Some people just want to lose weight because cow milk obviously contains more fat than plant-based milk, so it's not very ideal for some people in diets, or some people think it's not ideal. But I personally think that everything is good in moderation. And also, some people are just vegan. They don't want to drink milk from a cow. They're vegan. They don't believe in drinking milk from a cow. They want to drink non-dairy options. So there's many reasons as to why somebody might drink non-dairy milk compared to dairy. So we're just gonna go with lactose intolerance. Okay, moving on. Describe how a student can test a milk sample for glucose. Now I have a milk sample brought to me in the lab, and I want to test this sample for glucose. I want to know if this sample has glucose or not. What test could I do? I'm gonna do a test called Benedict's test, and I'm gonna add Benedict's reagent to the milk sample, and I'll heat that sample, and then I'll wait to see if there's any color change. I should see a red color change, or a final of a red color change. So we're gonna write that down. Add Benedict's solution to sample and heat. Color should change to red. Okay. Human breast milk contains special proteins that give immunity to the baby. Explain how these proteins can help protect the baby from diseases. So it's stating here that the breast milk that is fed to the baby contains very special specific proteins that normal milk doesn't contain, and these proteins sort of help build up the baby's immunity or give immunity to the baby. Now the question is asking you how these proteins can help that baby in protecting it from diseases. So these proteins specifically are going to be antibodies, because these are the proteins that help build immunity or protect your body from diseases. And these antibodies are specific, or they work against a specific antigen. Antigens are particles or things on viruses, bacteria, or pathogens in general, that antibodies recognize and bind to them. So these antibodies, basically, they recognize these antigens or specific to these antigens, and so once they see these antigens in your body, they start sticking or clumping onto them, and then destroying or killing these bacteria, breaking them down, phagocytosis, any of those methods. So for this question, we're gonna say the milk contains antibodies which are specific for certain antigens, and they stick to the pathogen and kill it, or actually kill the pathogen. Okay, that is the end of question 3. Okay, the peppered moth is found in many countries. 2 different forms of the peppered moth are a light-colored moth and a dark-colored moth. So we have this kind of moth, and it has 2 different variations. We have the light one and the dark one. The dark-colored moth was first observed in cities when pollution from burning coal stained tree trunks black. So they found these dark-colored moth in cities that had a lot of pollution, or where there was like a lot of coal being burnt, and this coal stained the trunks of the trees black. And so this is where they observed these dark colored moths. So it says scientists trapped moths in a city location from 1992 to 1998. The table shows the scientist result. We have a scientist that concluded the study. He trapped an amount of moth in a city from 1992 to 1998. We have the years on this side, and we have the number of light colored and number of dark colored moths. Calculate the difference between the percentage of moths that are dark colored in 1992 and the percentage of moths that are dark colored in 1998. So we want to know the percentage difference between the dark colored ones in 1992 and in 1998. Okay, so we're gonna calculate that by we're gonna get the percentage for the moth in 1992. Want to know the percentage of the dark colored moth in 1992 and the percentage of the dark colored moth in 1998. Okay, so first of all, you're gonna pull up a calculator. Please don't use your brain for this. We have the number for dark colored moth in 1992 was 27, and our total is gonna be the light plus the dark because we have both kinds in the city. So that's gonna be 27 plus 9, which is 36 times 100. Okay, so we're gonna do 27 divided by 36 times 100, which is 75%. So that is the percentage of the dark colored moths in 1992. Now in 1998, we had 9 and 13, so the total was 9 plus 13, which is 22. So we're going to do 9 divided by 22 times 100, which is 40.90, which is 41%. Okay, now I want to know the difference. So basically all you're going to do is you're going to deduct the 75 from the 41, and that is going to be 34%. Okay. Okay, for the next question, we have a graph, and we want to plot a line graph to show the number of light-colored moth and the number of dark-colored moth from 1992 and to 1998. Use a ruler to join the points with a straight line. So when you're doing any graph question for any IGCSE exam, you need to keep in mind a couple of things that you can do to get grades even if you don't know how to plot data. So first thing you want to do is you want to label the graphs right. You want to know which side is going to be the x, which side is going to go on the x-axis, and which side is going to go on the y-axis. Also you want to know how to like number the scale, how to scale the graph. That is also a very good thing to do. And also even if you don't know how to like plot correctly, you don't need to like plot the points exactly accurately, because there is a range for the plotting. So first thing, or a trick that I like to use for plotting graphs, I like to use, or I think this is common knowledge by now, but you need to know that everything on the left side is always going to be on the x-axis, and anything on the right side is always going to be on the y-axis. So for here, for this case, on the x-axis we're going to have the years, and for the y-axis we're going to have the number of moths. Okay, that is great. So we have from 1992 to 1998. We're gonna plot that here on the graph. So we have 1, 2, 3, 4, 5, 6, 7, 8. So 1992. We have all the way moving on to years. So 1993, 1994, 1995, 96, 97, 98. So Okay, so now we plotted our years. We're gonna plot the number of months. So when doing this, you want to look at your table and you want to look at the highest value you're given. So that's gonna be our top. So if you look here, we're gonna find that our highest value is 27. So we need to plot or scale the y-axis with our maximum being 30 or something close to that, because we don't want to make the scale too short. You want our top to be 30, so I'm gonna go with 5 every 2 large boxes, or 1 large box is gonna be we're gonna go up by 5s, so 10, 15, 20, 25, 30. You can do any order you like, as long as you're going with your scale above like half of it. So now that we have labeled our graph, we have done our scale, now we want to plot our data. Okay, so it wants us to plot our data for the light colored moth and the dark colored ones. So we're gonna go ahead and do that. So we have for 1992, 9 and 27. So we're gonna go ahead and do that. So I'm gonna go ahead and go 9 and 27. So 25 is gonna be in this area right here. You don't need to be really accurate with the sorry. Okay, I'm gonna do one with an X and one with a dot, and then we have for 993, 11 and 18. Okay, this is closer to 20, to be honest. 19, so I'm just gonna go down and scale it a bit. So that's 18, and then we have 11 and 7. So again, 11 here. Sorry, that's 1993. So 11 here. Sorry, and then we're gonna go down to 7. So it's gonna be somewhere in this region. Okay, and then we have 5 and 1 for the dark. Okay, so 5 and 1. So we're gonna go here, and we're gonna go to this area right here, and for the dark colored ones it's gonna be 1. So right here, actually that's too low, so it's gonna be here. Okay, and then we have for the light ones we have 1 and 4. And then 1997, 8 and 1. And then 13 and 9. Okay, now once you've plotted all your points, you're gonna go ahead and connect them with a ruler. So I'm gonna use this tool on the iPad because I don't have a ruler on here. So you're just gonna connect them. Okay, and then here we're gonna do the same thing. Okay, and then once you have connected them, you can label them. So I'm gonna go ahead and label them. I'm gonna go and say this is light colored, and then this is dark colored. Okay, this is how you plot a graph. So let's move on. Okay, so in the 1990s, many cities introduced laws that prevented the burning of coal. Comment on the changes in the number of light-colored moth and number of dark-colored moth between 1992 and 1998. So they're saying that in the 1900s, in the many cities made laws that stopped companies or manufacturers or factories from burning coal. So it wants you to comment on the differences in the light colored moths and dark colored moths from 1992 to 1998. So we're gonna go ahead. You don't really need to look at the graph if you don't want to. You can just go ahead and look at the table. So I'm gonna start with the with my commenting on the light ones, and then I'm gonna move on to the dark ones. So as you can see that overall from 1992 to 1998, the number of the light colored ones sort of increases. So we start at 9 and then we end at 13. So we have an increase. And if you look, we can do a side-by-side comparison. If you look at the dark or the number of the dark colored moth, you can see that we start at 27 and we end at 9, so there's an overall decrease in the dark colored moths. And overall, like the overall total number of the moth is decreases in both light and dark. The total is decreased overall. And okay, so we want why. Okay, now we're gonna go by year. Obviously, in 1992 we had more light colored moth than we did dark, and moving on to 1993 we had the same situation. But in 1994 it looked different for the light colored ones. The light colored ones were so much higher than than the dark colored ones. So at the start of 1994, or at the end of 1994, we had more light colored moth than we did dark. But before that, we always had more dark ones. So this could be maybe to the less coal being burnt. Maybe there's less burning of coal, as we did state in the question in the beginning. It stated that the dark colored moth were because of the burning of the coal, which stained the tree trunks. Maybe there's less dark colored ones because there's less coal being burned by manufacturers or companies. So yeah, okay, so we're gonna go ahead and write all these points down. We have written our answer. We are gonna move on to the next question. This is the end of question 4. This is part 1 of the video. I will continue the rest of the half of the exam in part 2. I would like to thank you all guys for watching till the very end, and this is Ace IGCSEs with Clarissa, and I hope you like this video and subscribe to this channel and tune in for the for part 2 of the video. Thank you so much.